From California, San Diego, I'm going to talk about phylogenetics and genome-wide phylogenetics. It's more of a tutorial. Most of it is not my own work. I'm just giving you some sort of background information. Everyone knows what a phylogeny is here. It's just a tree that shows the relationship between different species, and it has been creeping up in various talks. So I don't have a slide on why phylogenetics is important, just because every one of you have already talked about phylogeny one way or another in your talks. I think that's a good enough reason. One quick point I want to make here is that a phylogeny is not just a description of what two genomes are closer to each other in distance. It's actually a biological hypothesis. We are making this phylogeny is making the hypothesis that humans and chimp had a common ancestor that the other species didn't share. So it's not just a computational object, it has biological meaning. Okay, so how do we go about inferring phylogenies? These days it's mostly from genetic data, so you get your genetic data of interest from your species of interest. The DNA will not match, but they will be similar. And the reason we can use the DNA to recover the phylogeny is because their similarities and differences are a reflection of the phylogeny. So the common ancestor had some sequence, mutations happened over the phylogeny, and now you have the data. Obviously, you don't get to see the phylogeny, you just see your data. So you have a data matrix like this with various sites. Each site is supposed to be homologous, meaning that all these A's are supposed to have a common ancestry. And you want to get to the phylogeny. This is, again, the same phylogeny, just drawn unrooted. It's a tree, unrooted tree. And sort of the standard way of doing, sort of going from the data to phylogeny these days is using likelihood. There are a ton of different methods as well. I'm not going to go through them much right now, maybe a little bit later, but the standard way from genetic data to phylogeny is likelihood. So how does this likelihood framework work? Oh, before I go there, another thing that we could do if we have a likelihood framework is to also estimate sort of how much confidence we have in various branches of that phylogeny. So in this case, we are saying, so when I say statistical support, this is what I mean. How much confidence we have in any branch of the phylogeny. Okay, so how does this likelihood work? It has really three.
Very basic question. Do you look at windows or which pieces do you compare? How big they should be?
So I'll get that in 1 second. Very good question, but I'll get into that. So right now, I'm just assuming I guess you're talking about the data. So I'm assuming you, as the biologist in the room, has found some gene that they're interested in. We are looking at that. But most of my talk is going to be about that question. Okay, so how does the likelihood work? It has three main ideas. Before I get to the three main ideas, it's not so much an idea. It's just an assumption. This first thing to know is that we usually assume these sites are independent. Right? So if you have, whether 10 sites in your alignment, site 1, site 2, site 3, we assume are independent. Not that they are, but that's what we assume. So the likelihood of your tree can be computed sort of as the product of the likelihood of different sites. Okay, so with that assumption, you need only three ideas to be able to compute the likelihood of a tree given your sequence data. The first one is this idea of using a continuous time Markov chain for a branch. So if you have a branch of length t, you start with some letter, let's say C, and you wind up with another letter G. You can compute what's the sort of likelihood of what's the probability of this event happening? What's the probability of going from C to G in time t? So for this, we have continuous time Markov chain models of sequence evolution. The second idea is quite intuitive, which is if you have a tree like this, and if you had the labels of internal branches, internal nodes, if you had the labels on the internal nodes, then the sort of evolution of branches would become conditionally independent, meaning that conditioned on this being C, what happens here is independent from what happens there. It's just because you have a tree. That's the second idea, quite intuitive. The third thing is obviously we do not have these internal labels. So what do? Back in the days, in I think 80s or something, an algorithm was developed by Felsenstein's. It's called Felsenstein's pruning algorithm, which enables you to kind of sum over all possibilities for internal branches. It's not too different from what you would do for a hidden Markov model, for example. It's a very similar sort of dynamic program. With this sort of three pieces and that assumption put together, we can compute the likelihood of any given tree for any given sequence data. And there are like a lot of details that I'm not going into here. It's just a basic idea. Now, what I want to emphasize here is that this is like any likelihood, other sort of a statistical inference. So this is kind of just like you have a coin you're tossing. It has some bias. You want to know what's the bias of the coin. You toss it 100 times. You can try to estimate it. If you now toss it 1,000 times, you get a better estimate of the bias, right? So you want the sort of the bias of the coin corresponds to the tree here. What corresponds to the number of times I'm tossing the coin? Anyone? Not number of the sequences. Number of sequences does not give you more information. It's the length of the sequences, right? How many sides do you have? So in this case, we have 10. It's like I'm tossing my coin 10 times. If I had 100 sides, it would have been I would have had a better estimate, right? So this takes me to the next slide, which is this idea of having phylogenomics. Phylogenomics is basically saying traditionally people used to build phylogenies from individual genes. Now let's use the entire genome. Why? Because of exactly what I just said, which is if you have the entire genome, it's like you are throwing your coin 3 billion times as opposed to 1,000 times. It's supposed to give you a better statistical estimate. So the idea was more data, better, sort of less error in your estimation. So when phylogenomics came about in early 2000s, people wrote papers saying that phylogenetics has always been a very contentious field. There has always been difficulties in terms of, is this the correct tree? Is that the correct tree? And the hope in early 2000s was that we are getting over that. Having the entire genomes is going to be the end of incongruence. Incongruence is this idea that when you get different analysis of the same sort of group, you get different trees. Okay, so we hoped it's going to be the end of incongruence. You can all imagine what happened. Instead, it was the beginning of incongruence. It was just a few years later that people wrote papers saying that phylogenomics is not the end, but the beginning of incongruence, which has actually panned out to be the case. Phylogenetic has brought with it a lot of incongruence. Okay? So now I want to just talk about why that's the case. That idea of having more data, less variance in your inferences is still a correct idea. But why is it that it fails? There are many reasons. The most important or maybe more interesting one is the idea of gene tree discordance. So gene tree discordance is this. You have your 4 species of interest. You go across the genome. You look at one gene and you look at another gene. And I think I forgot to mention this, so let me just say that right now. When I talk about genes here, I'm not talking about functional genes. I'm not talking about protein encoding genes. I'm just talking about some region of the genome, which is hopefully recombination free, but it's basically any stretch of the genome that's small enough, without getting into the weeds of what actually it means. Okay, so back to the idea of gene tree discordance. As you go across the genome, the true phylogenetic history can change from one part to another, right? So for one part, you can have gorilla and human be sister to each other. For another part, you can have human and chimp to be together. Does this mean it's hopeless to think about one tree? No, it doesn't. This is very important to understand. Even though you can have phylogenetic discordance, in some cases, not always, in many cases, it's still meaningful to think about one tree that sort of captured the entire genome. we are going to call that the species tree, and the tree for each region, we call it the gene tree. Why is it still meaningful to talk about the species tree? It goes back to the point I made in the first slide. The species tree is not just a it's not just something you get from your computational method. It's a real hypothesis about how evolution happened, right? So if your hypothesis, the species tree is making the hypothesis that there was a real population of apes that from which humans and chimps originated, but gorillas did not, right? So no matter what's happening in your genome, that's still meaningful, and we'll see sort of more mathematically why this is the case. Okay, so now the question is, why is this happening? Why can you have different gene trees for different parts of the different parts of the genome? And it turns out there are many, many different reasons. 3 or 4 main ones that I'm going to quickly mention, and then maybe focus on one. Question? No? Anyone else? Any questions so far? Okay. So, reasons of discordance. First one, duplication and loss. So for a lot of species like plants, you have rampant duplication and loss of genes. And so when the gene gets duplicated, the 2 copies are called paralogs. And when 2 genes are in 2 different species are related to each other through just speciation events, they're called orthologs. And so this is, so the gray tree here is your species tree. You had a duplication here. Now you have 2 copies of that gene, the blue copy, the red copy. Every species has 2 of them. This would be your gene tree, which obviously is not the same as the species tree because you have more leaves. So far, it's not even so interesting. But it could become even more interesting when you start also adding losses. Okay, so here I have 1 duplication and 3 losses. And I get this tree, right? And you don't get to see the colors, right? So imagine you don't know the red and blue color. Then you could, so you have a gene tree now that puts 3 and 1, the species 3 and 1 together to the exclusion of 2, but in your species tree it's the other way around, right? So with enough duplications and losses, you can create scenarios like this where the gene tree and the species tree are incongruent. It doesn't even have to be losses. It could be just that you didn't sample it, right? So maybe these were not lost, you just didn't sample them. Okay, so duplications can give you differences between gene trees and a species tree. We have a statistical models for sort of generating these kinds of gene trees given the species tree. And those models are mostly based on birthless processes. I'll maybe say a little bit more about that later. Any question about this part? Okay, so the next thing is horizontal gene transfer, which is something a lot of you guys who work on microbiome have thought about more than I have. But so I guess this one is a lot more clear, right? So you have your species evolution going on, but this 1 gene out of a sudden in B is inherited from C, not through vertical inheritance, but by transfer. So it just picks it up. It picks up that genetic material from the environment. This is horizontal gene transfer. It's rampant in prokaryotes, and it's observed in eukaryotes. How much it happens in eukaryotes, I don't know. I don't think we know in general. And when you have these events happening, a tree for the species phylogeny may be insufficient. And so now, whether it's insufficient or not, I guess it's a matter of debate. I'm not going to get into that debate. It probably is a matter of degrees, how much of it is happening. Okay, so some interesting points that I want to just make sure everyone keeps in mind about HGT. When you have this event happening, you get the new gene. A lot of times the target, in this case B, also has a gene similar to this gene, right? So now you might have 2 copies of it. But it could also be not the case, right? So it could be that the target species will have something similar, so now you have 2 or not. Another thing that is a little bit more problematic when we get to the modeling is that this is a nice scenario where the donor this branch has a descendant in our sample or today, right? But a lot of times, whoever donated this genetic material is either extinct, meaning we don't have any descendants from that branch, right? So you can't draw these nice lines from one branch to another, given your current phylogeny. Or it might not even be extinct, but you just haven't sampled it, right? So you could have like this dangling transfers from an unknown source. And so modeling that is difficult. There are various statistical models that have been developed to study HCT, including by some people in this room. Saigi has one with Sebastian Rock. Others have models. There are several models that can be used. Okay, so it's close to horizontal gene transfer is the idea of hybridization. It's not the same, but it has some similar features. And hybridization is when you have two species that are supposed to be different species, but somehow they manage to produce offsprings that are viable, right? So now you have a species that has parts of its genome coming from one species, in this case, species B. Parts of its genome is coming from an ancestor of A. Parts of its genome is coming from an ancestor of C. And so B is a hybrid species. Obviously, when you have hybridization, some of your genes, like the yellow one, is going to be, your gene trees are going to be different, right? Some of them are going to follow that path, others are going to follow this path, so you get differences between gene trees. How much hybridization happens is not very clear. We definitely know that it happens. How rampant it is, I don't think we know yet. So it's an important thing. Now, when you have hybridization, it can lead to some philosophical questions that I'm going to just mention, but not go into any detail. At that point, you have to ask yourself, what did the species even mean to begin with? If they can, why were we calling C and A two different species if they could have produced offsprings? And there are various ways of defining a species and finding a species. All of this is very much up for debate and up for methods development as well. Okay, so this is the last one, the one that I work on most and is maybe most interesting to people who do population genetics, incomplete lineage sorting. So this has already been mentioned. Let me just repeat what it is. Incomplete lineage sorting is sort of what you can get from coalescent not happening. So look at that red population that is ancestor to human and chimp. In this blue gene tree that I have drawn here, everything is happening fine. So you have two lineages coming up, one from human, one from chimp, and they are very happily coalescing inside that population, no problem. But it's, coalescence is a random process. It could have happened that they didn't coalesce, right? So let me just go back and forth so that everyone sees this, right? So just by random chance, the coalescence could have failed to happen in that red population. And when the coalescence doesn't happen there, you go further up. Now you have three lineages, and human has the same chance of coalescing with gorilla first, or with chimpanzees first, or there's 1/3, 1/3, 1/3 chance of that each of the two possible coalescences, or each of the three possible coalescences happen first. And in this case, human is coalescing with gorilla before it coalesces with chimp, right? So this is what incomplete lineage sorting is. If I remember the estimates correctly, it's about 70% of our genome, we are closer to chimp. In another 15%, we are closer to the gorilla. And in the remaining 15%, gorilla and chimp are closer to each other than either is to human. Okay, so what's the likelihood of this lack of coalescence? We call it deep coalescences sometimes. It's basically what's the length of this branch, how many generations you have. Obviously, the more generations you have, the less likely it is that two things fail to coalesce. And also, what's the population size? Right? The bigger the population size, the more likely that lack of coalescence or deep coalescences happen. We have a statistical model to study incomplete lineage sorting. It's called multi-species coalescent model. It just takes the sort of traditional Kingsman coalescent model, and now you have a tree of different populations. Whatever fails to coalesce in that population gets inherited in the parent population. One important thing about the multi-species coalescent model is that under this model, if you give me the species tree and all the branch lengths and population sizes, it defines a unique distribution on the gene tree topologies, and the opposite. It's identifiable, meaning that a gene tree distribution corresponds uniquely to one species tree topology. So we have some hope of recovering the tree despite all the differences between gene trees. Okay, so let me just recap what we learned so far. So far we learned that gene trees can be discordant for 4 different reasons. But now I guess the obvious question is, so what do we do with that? How do we take, we have to take in account of it, but how? There are 4 main ways in which the literature has, there are 4 main questions that you might be interested in. One is that you have a species tree that you have estimated, you have a gene tree that you have estimated, you want to map them together. This is called reconciliation, mapping a gene tree to a species tree. And this is what I have been doing so far with my figures, right? All these figures, when you have a gene tree inside a species tree, that's reconciliation. The second thing you might be interested in is inferring a species tree given a collection of gene trees, right? So I have my whole genome, I've chopped it up into small segments. I get a gene tree for each region using, let's say, the maximum likelihood procedure that I described. Now you want to combine all of them to get a species tree. The input is a bunch of trees, the output is a tree. This is a goldmine of computational problems. It's something I have worked on, many other people work on as well. There is also the opposite problem, which is also very important, less widely recognized, which is you have an estimate of the species tree that you think is accurate, and you are interested in the phylogeny of one gene only. Maybe it's a gene that has some functional importance to your study. When we infer the phylogeny of a gene, it's going to be inferred from a limited length, right? So you are throwing your coin, let's say, 1,000 times. So it's going to be noisy. But remember that the gene tree is evolving inside the species tree. So if you have the knowledge of a species tree, you can try to use that to improve your estimate of the gene tree. This is something sometimes called tree fixing. So the input is a sequence data for one gene and a species tree. You try to improve your estimate of the gene tree compared to if you had only the sequence data. The fourth question that comes up is co-estimating gene trees and the species trees, right? So if you have just sequence data, you don't know the gene trees, you don't know the species tree, can you co-estimate all of them in one statistical analysis? This is what everyone wants. It's very difficult to do with large data sets. There are other problems also that I'm not sort of mentioning here. There are 3 main approaches that people have used to address each of those 4 questions. Parsimony-based, model-based, summary-based. So let me just tell you a little bit about each of these approaches. The parsimony-based methods are based on the idea that if there are some events that happen that lead the gene tree and the species tree to be different, can we minimize the number of such events that we need to explain our gene trees and the species tree? So an example is shown here where your species tree is the thing shown in blue, and what you have in green is the gene tree, right? And we are sort of mapping the gene tree to a species tree using one duplication event over here, right? And a bunch of losses, 1, 2, 3 losses. So that's one reconciliation of your gene tree with the species tree that requires 1, 2, 3, 4 events. Is this the most parsimonious way of mapping the gene tree to a species tree? No, there is another one that requires fewer events, right? So this has one duplication and 2 losses, so it's more parsimonious. Finding these most parsimonious reconciliations is really, really fast. It's linear time. And that's why sort of doing all sorts of downstream analysis based on parsimony is very efficient. That's why people use it. The problem, obviously, is that if the rate of duplication and loss is high enough, the most parsimonious answer is not the best answer.
So this is, this is polynomial.
Finding the most parsimonious mapping is linear time, I think. Definitely polynomial, it's either linear or quadratic. It's very, very fast. I think it's linear.
And of course, so this is a reconciliation between, you have both guys.
Yes. 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 But so if you want to now find the most parsimonious species tree, given a bunch of gene trees, is not polynomial anymore, but at least computing the score is polynomial. Whereas when we get to the model-based stuff, even computing the score is not polynomial. Okay, so actually let's get to the model-based stuff. So if you're not doing parsimony, you're doing statistical models, and the statistical models used in this field all follow this hierarchical approach. So it's a hierarchical statistical model. Sort of your parameter, you can call it the hyperparameter here, is the species tree. That species tree with some model of gene evolution defines a distribution over trees. Right? So you need a model that, given a tree, defines a distribution over trees, which are your gene trees. Right? This we are going to call gene evolution model. And then you need sequence evolution model, which is, given each of these gene trees, you need a model to generate sequence data. Right? So we have a third of these 2 steps. And if you want to now, so this is the generative model. If you want to do inference, you obviously go the other way around. And so you could, one thing you could do is sort of do each of these inferences one at a time, right? So get a bunch of gene trees and then combine them to get the species tree. This is called the 2-step approach. Or you can try to do inference under this sort of very complicated hierarchical model in one go. That's what co-estimation is. There are, like I said, there are various models. I'm going to go a little bit faster. There are various models of gene evolution. For ILS, we have multi-species coalescent model. For duplication and loss, we have these birth-death models where birth corresponds to duplication, death corresponds to loss. For horizontal gene transfer and hybridization, we have these sort of network DAG-based models. And importantly, we have also some models that combine multiple causes of discordance. For example, if you're thinking about hybridization, you should also be thinking about ILS. You can't sort of think about one and not the other. You will be misled, right? But there are models that combine these. The problem is, once you build these models, inference under all of them becomes extremely difficult. So even computing the likelihood of a species tree for a given gene tree is often NP hard. So we don't really have good ways of doing inference under many of these models. Not all of them, but most of them. So that leaves us with summary-based methods. Summary-based methods are methods that sort of try to avoid making that computation, but remain statistically consistent under those models. So you don't compute likelihood, but you stay statistically consistent. So let me just give you an example of how this would work. So under that multi-species coalescent model that I mentioned, if you start with only 4 species, and you are thinking about unrooted trees, you have only 3 possibilities. And we have this result that says the most probable gene tree always will match the species tree. for 4 species. Okay, so the gene tree that has the highest probability matches the species tree. If you know this, then your maximum likelihood estimate of the species tree is just the most frequent gene tree, right? That's very simple to estimate. You just, for your 4 species, you just get a bunch of gene trees, you see which one happens more often. That's your estimate of the species tree. The problem is when you go to more than 4 species, these results don't hold up anymore, right? So now you can have situations that are called anomaly zone, where your most frequent gene tree is, or most probable gene tree is different from the species tree. So you can't just list all the gene trees, count them up, and take the most frequent one and say this is my species tree. What can you do instead is play a trick. Play a very simple trick. If you have 5 species, you divide your data set into all n choose 4 possible quartets. For each of those quartets, the previous results is still correct, right? So for each of them, you can just count up the number of times you have seen each of the quartets. Now you have, and so you have an estimate of what the tree should be for each of those n choose 4 quartets. And then you can combine them. This is a method that several methods, this is an intuition that several methods have used. And then we had a very simple intuition that instead of, for each of these n choose 4 quartets, if instead of picking the best, the most frequent one, if you just weight all the n choose 4 quartet topologies multiplied by 3, if you just weight all of them by their frequency, and find the tree that matches sort of this weighted matrix, that remains a statistically consistent estimator of your species tree. So let me just rephrase that. We get to this maximum quartet support the species tree problem, where your input is a bunch of gene trees. Each gene tree defines a set of quartet topologies, n choose 4. Each species tree defines a set of n choose 4 quartet topologies. And you can see what's the intersection for each quartet. And the score of your species tree is just this number, summing over all summing over all gene trees, right? And this, if you could solve this problem, it would be a statistically consistent estimator of the species tree. The problem turns out to be NP hard, which is not even surprising because everything in phylogenetics is NP hard. Okay, so yeah.
What was M?
M was the number of gene trees. M is the number of gene trees, and I'm going to change my notations very quickly. So I equals 1 to.
Goes from 1 to M and T sub I. T capital.
This is, yeah, that's one gene tree. Yeah, so you have MG.
Oh, this T is sub I. Okay.
Yeah, so it's basically the total number of quartet topologies in all the gene trees that match the species tree. It's what you would have called weighted quartet score, or what you have called the weighted quartet score in your papers, right?
So it's max over capital T. Okay.
Yeah, so this is the score of a T, and then the species tree would be the tree that maximizes this score. Okay, so we developed the dynamic programming to try to solve it. To solve the problem is obviously exponential running time because the problem is NP hard. We came up with a constrained version of the problem. So it basically boils down to not searching the entire search space, searching parts of the search space that are defined by or input gene trees, and this becomes polynomial time. Don't forget, don't worry about what exactly that polynomial is. It becomes polynomial time, roughly quadratic in practice, and this constrained version you could prove remains statistically consistent, and we could also sort of drive equations showing how many gene trees do you need before this method converges to the true species tree with high probability. So I have some results about Astral. I'm going to just skip over them because I don't want to go over the time a lot. It's just showing that in simulations with large number of species and gene trees, it works very well. And we had some more results I'm going to skip over that basically said even though the method is designed for ILS, in simulations it also works very well when we have horizontal gene transfer, including if we have a lot of horizontal gene transfer. So let me just skip over all of that and tell you where we are right now with Astral. So this tree right there is a tree we recently inferred and we haven't published yet. It's a tree that we inferred from 10,000 bacterial and archaeal, so microbial species, sampled from the 100,000 that are available right now. But this is based on 400 genes. So 400 genes at 10,000 species, that's the kind of data set size that we are dealing with right now. And with parallelism and GPU and things like that, this can be handled. This size of data set can be handled presently with the methods that exist. Okay, so now I want to switch and very quickly talk about data.
Can you go back to the Can you circle the eukaryotes, right?
Oh, we don't have eukaryotes. It's only bacteria and archaea.
Are you planning to include methylgenome and genome?
Yes and no. I mean, so we probably will place them, but yeah. So it's maybe. Let's just say maybe.
The reason I'm asking is because there's a very fundamental discussion going on here regarding how to place archaeal branches with new ASCAR genomes that are emerging.
Right. So we do have this CPR group here, but yeah, but we don't have the all the metagenome sample. That's once we get this out of the way, then we'll think about whether we want to get into that. There was a more question.
When you started, you assumed that different genes are independent, but in bacteria, one unit, but no combination.
So, I mean, again, I'd love to hear about this from biologists in the group, but my understanding is that the rate of recombination in bacteria is very high. It's not lower than necessarily eukaryotes, even though it's asexual. Other mechanisms can give you recombination, and the overall rate of recombination, I've seen papers making the claim that it's about the same as eukaryotes. I'd be interested to learn more about that. But maybe if the discussion is about that, maybe we'll.
I just had one other thing, which is when you're using all the genes, different gene families evolve at different rates in different branches. And
so that comes very natural in this framework because each gene is inferred separately. They're allowed to have their own rate.
But you have different branches as well?
Yeah, every gene tree is just inferred independently, right? So they can just have whatever rate they have.
No, I mean like in one phyla, some gene families will move very quickly.
Yep. And some won't, right? And so there's no problem with because your gene trees are inferred independently again, right? When you're inferring gene 1, you don't look at the other genes.
But you're weighing I assume you're weighing the
When we get to the species tree inference, we are only looking at the topology of the input gene trees. It's only like the topology of the gene trees gives you the topology of the species. But maybe I'll move on and then we'll take that offline. Okay, so what kind of data do we use? People have, if you have whole genomes, you can use that. So if you have whole genomes, you could use that. If you have, most often we don't. We go for sort of samples of whole genomes. It might be transcriptomes, which are not random samples, but they're samples. People use targeted capture, where you have a special sort of libraries of loci that and you target those. Well, let's leave this for phylogenetic people. But one of the more interesting developments that's very recent in this field is genome skimming. So I want to just quickly tell you about genome skimming. So genome skimming is this idea that you have your genome of interest. Instead of generating data enough so that you can assemble it, you just get very low coverage you just do shotgun sequencing at very low coverage, not enough to get assembly. Think 1x or 2x or something. Most people are doing genome skimming to answer questions about, so basically to replace traditional barcoding. That's the main use. And so this is something we are starting to look at, but genome skims can also be used for phylogenetics. And this is a claim I'm making. It's a new thing. We'll see how it pans out in near future. But I think it will be a new way of doing phylogenetics that's very interesting. So it will be distance-based phylogenetics probably because you can't assemble. And the way it could work is the following. You look at all the k-mers and all the reads from, let's say, 2 sets of genome schemes. You compute the Jacquard index between the k-mer sets. The Jacquard index can then be translated to Hamming distance. And this is something that has been known for a while. And Adam Flipper's lab also used this in MASH. You get the Hamming distance. And once you have the Hamming distance, you can try to translate it to phylogenetic distance. These formulas are all in the vanilla case. If you make a ton of assumptions and everything is vanilla, all of these will work. Okay, so let's do some simulations. We took some insect genome. We added 5% distance to it by just showing mutations added. And we then skimmed it with coverage that ranged from 1/4x to 16x, right? Then we computed the distance using the approach I just described. And as the coverage goes up, eventually your distance becomes very accurate. You get a very accurate estimate of the distance. But you need something like 8x coverage for this particular case. And so what Shahab, who is here, also did was he sort of took that formula that translated Jacquard to Hamming distance and updated that so that it can take care of low coverage as well. Sort of has a way of estimating coverage and taking coverage into account. We made a method that we presented last year in Recom. It's called skimmer. And skimmer basically, it's the same figure now, but even at low coverages, you get very good estimates of distances. And if you go, and Metin, who is also here, is now working on improving the translation from the Hamming distance D to phylogenetic distance T, which I showed in the previous slide. But even this simple version of it, if you apply it to a bunch of Anopheles genomes, you skim them at very low coverage, you get a tree that matches pretty well with the sort of the reference tree from Open Tree of Life. But if you just look at the marker barcode genes, that will not match a lot. All those red branches are branches that don't match. Okay, so I had some more stuff, but I will skip them. So I had the so far slide, and I wanted to keep going, but I'll probably just stop there. So let me just use this as a summary slide. So I guess just to summarize, what we saw was that there are sort of 3 or 4 main types of discordance between gene trees and species trees. There are 3 main approaches that you can use to try to infer relationships between gene trees and species tree based on parsimony model, statistical models or summary statistics. There are 4 main questions. You might be interested in reconciliation, species tree estimation, gene tree estimation, or co-estimation. And there are at least 3 types of data: whole genome data, subsamples of the genome, or more recently, these genome schemes. And it remains to be seen in near future if these genome schemes, which are a lot cheaper to get, can replace sort of some of these other types of data. It may or may not be able to do that. We'll see. So that's it.
Results on genome skim depend on the length that you have. So you showed results like 2 or 16x coverage. Is that like for a megabase? Or
So that particular genome was, I think, a couple of 200 megabases. We started with the genome of an insect that was 200 megabase. I think what matters more, you can, like in theory, what matters more is the coverage rather than the actual length, because the Jacquard index you can show is a function of your true phylogenetic distance plus coverage. If your coverage is high enough that every place gets sampled at least once, or close to at least once, then the coverage doesn't matter. But if your coverage is low enough, the intersection could go down, the sort of the numerator of the Jacquard can go down either because of phylogenetic distance or lack of coverage. So no matter what the length, the coverage still is the important factor.
So in your gene tree, when you're just going through the genome, I guess you're doing whole genome alignment and then finding sections to cut them into fixed length and mutation.
That's one way to do it. If you are lucky enough to have whole genomes, you could do that. Oftentimes, that's not what we do. That's my favorite way of doing it. Most times, we wouldn't be doing it that way. People start with gene families that, from previous knowledge, we know this is a gene family, this is another gene family. Usually, people start with that knowledge and take it from there. But if you afforded to do that, yes, that would be very good. There is no constraint about like frames or anything. You could use.
What I was wondering, because then you are in fact looking at positions that are interesting and are not necessarily on the evolution of how these things are.
The other way around. So the less selection you have, the better for phylogenies, right? So if you're trying to answer questions about selection, no. But if you're trying to infer the phylogenetic relationships, anything that biologically is meaningful is hurtful. You don't want any real biology. You want places that are evolving neutrally with no real sort of nothing interesting going on, because the models of evolution that we are using assume that. They assume a neutral random evolution.
All right, let's take a break till 2:45.